Next up is post-pickling processes. What can preservation fluid chemistry tell us about protein survival in wet collections? By Tiffany Shea Slater at the University College Cork.
Hi, everyone. My name is Shay, and I'm a postdoctoral researcher down at UCC in Cork, and I'm a soft tissue taphonomist. So my research primarily focuses on the preservation of proteins and other biomolecules in the fossil record, but I'm really excited to start looking at younger samples that are preserved in spirit that may inform on our biodiversity crisis. So the use of wet collections for genomic research is increasing, which is really exciting, but proteins also offer critical information about biodiversity loss and especially physiological responses to climate change. Yet these have received a lot much less attention for various reasons. And one of those is because of a lack of understanding about post-pickling protein taphonomy. So alcohols and formalin they're not chemically inert, and they can dehydrate and precipitate proteins, but the fluids themselves can also oxidize, evaporate, and become contaminated. So as a result of this, proteins, lipids, and other biomolecules may leach into the surrounding fluid. And this is compounded by the fact that in many historical collections, we have poor documentation of the specimen history, the fluid identity, concentration, and especially the maintenance history. So this creates a major problem for both collections conservation and also research. So the aims for this pilot study are to determine whether proteins can actually be preserved in preservation fluid, and to identify the potential controls on fluid chemistry and potentially protein preservation, such as taxonomy, evaporation, fluid type, and age. So this will also identify key variables to investigate in future controlled taphonomic experiments, which will enable extensive destructive sampling, which of course we don't want to do to our collection. And of course, this will aim to support the long-term care of spirit collections at UCC and hopefully elsewhere. So the aim here will be to sample the fluid and identify any potential proteins or lipids that are in them as a proxy for the state of degradation of the specimen. And to do this, I'll use FTIR and Raman spectroscopy, which are really great, require very minimal sample amounts. So about 41 jars have been identified to represent distinct taxonomic groups, and these all contrast in their overall tissue chemistry. So there are also opportunities within each group to identify other potential controls on preservation. So our soft-bodied cephalopods here, these are there's a number of these that have been accessioned sequentially and maybe have entered the collection around the same time. But the interesting thing is that they really vary in their condition. So we have relatively pristine specimens there at the top, and then we also have some that have a considerable amount of sloughing and cloudiness, and they differ in their fluid colors. Then we have our lampreys, and this is perhaps the most controlled group out of all of these. And they're all similar in age. They're all from, I think it's 1973. They're similar in their fluid clarity, but importantly, they have a gradient of fill levels and thus specimen exposure. So this can serve as a really nice natural experiment to examine the impact of evaporation on long-term preservation. And then we have our fish. So this is the most diverse in these four groups. So they're really diverse in their terms of age and their fluid clarity, the specimen exposure, and especially the species diversity. But I have highlighted here just a few in this group that are ecologically important and, of course, interesting to study here. And then lastly, we have our chondrichthyans. So we have a lot of variation in this group as well, but they do seem to have darker fluids than the other groups here. So this might be a really unique opportunity to study whether the very distinct protein and lipid profile of chondrichthyans has any impact on fluid chemistry, because they are very distinct to the other groups presented here. And again, I've just highlighted a few that are of really real ecological importance. And most importantly, each of these groups has at least one historic specimen, with all except in the chondrichthyans having a species equivalent for a modern, a modern specimen. So that enables a really nice direct comparison, and they're likely to be preserved in different fluids, so it may produce some interesting comparative data there. I'm also really happy to discuss all of the different variables that are involved in this later. And really, the hope is that all of this will identify preservation conditions and specimens that are compatible for future proteomic research. And of course, we want to help position natural science collections as real active resources for addressing contemporary biodiversity questions. And I also want to point out that this is part of a wider effort to rejuvenate the historical collections at UCC. And please check out Fidelma's poster to see more highlights in our collection. So thank you very much.